What is the recent development to gene therapy and stem cell research?
What is gene therapy? Gene therapy is when DNA is introduced into patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effect of a disease-causing mutation.
Gene therapy uses sections of DNA to treat or prevent disease.
The DNA is carefully selected to correct the effect of mutated gene that is causing disease.
The technique was first developed in 1972 but had limited success in treating human diseases.
Gene therapy may be a promising treatment option for some genetic diseases including muscular dystrophy and cystic fibrosis.
What is stem cell research?
Stem cell research is the area of research that studies the properties of stem cells and their potential use in medicine. As stem cells are the source of all tissues, understanding their properties helps in our understanding of the healthy and diseased bodies develop in homeostasis.
Where do stem cells come from?
There are several sources of stem cells. Embryonic stem cells. There are stem cells came from embryos that are 3 to 5 days old. At this stage, an embryo is called a blastocyst and has about 150 cells. Adult stem cells. These stem cells are found in small numbers in most adult tissues, such as bone marrow or fat. Compared with embryonic stem cells, adult stem cells have a more limited to give rise to various cells of the body.
Adult cells altered to have properties in embryonic stem cell. Scientists have successfully transformed regular adult cells into stem cells using genetic reprogramming. By altering the genes in the adult cells, researchers have programmed the cells to act similarly to embryonic stem cells. Prenatal stem cells. Researchers have discovered stem cells in amniotic fluid as well as umbilical cord blood. These stem cells have the ability to change into specialized cells.
And now we are done defining what is gene therapy and stem cells. Let's move on to the recent development of gene therapy and stem cells research. Stem cell grafts and rehabilitation combine boost spinal cord injury results.
Animal models, researchers found that intensive physical therapy in tandem with implanted neural stem cells increased tissue growth, repair and functionality in spinal cord injuries more than those treatments alone.
In a new paper published August 22, 2022 in a journal GCI Insights, researchers at University of California San Diego School of Medicine addressed the question of whether rehabilitation can augment functional outcomes when combined with pro-regenerative therapies, such as stem cell grafting. Using a rat model, researchers induced a cervical lesion that impaired the animal's ability to grasp with its forelimbs.
There were 4 groups: animals who underwent the lesion alone, animals who received a subsequent grafting of neural stem cells designed to grow and connect with existing nerves, animals who received rehabilitation only, and animals who received both stem cells therapy and rehabilitation. Rehabilitation therapy for some animals began 1 month after initial injury, a time point that approximates when most human patients are admitted to SCI rehabilitation.
Rehabilitations consisted of daily activities that rewarded them with food pellets if they performed grasping skills. The researchers found that the rehabilitations enhanced regeneration of injured corticospinal axons at the lesion site in rats. and the combinations of rehabilitations and grafting produce significant recovery, and forelimb grasping with both treatments occurred 1 month after injury.
Gene therapy targeting overactive brain cells can treat neurological disorder.
No treatment for neurological and psychiatric disease that works by reducing the accessibility of overactive brain cells has been developed. Many brain diseases such as epilepsy are caused by excessive activity of a small number of brain cells. These conditions often do not respond well to drug treatment, mainly because drugs affect the whole brain.
Whilst genetic therapies could be a promising way to treat these conditions, current methods do not distinguish between overactive and normal brain cells. However, the new treatment outlined in Science and tested in mice uses a technique that only alters overactive cells and spares those that are acting normally. Corresponding author Dr. Gabriel Lignani, or UCL Queen Square Institute of Neurology, said, "We invented a gene therapy that switches on only in overactive cells and switches itself off if activity returns to normal. We harness the ability of certain DNA sequences to control gene expression in response to metabolic signals."
By redirecting this activity sensing mechanism to drive the production of molecules that stop brain cells from firing, we show that epileptic seizures can be suppressed. To create the gene therapy, the team screened several genes known to switch on in response to stimulation and coupled their promoters, DNA sequences that determine whether the DNA is copied to RNA, to potassium channel chosen for their ability to reduce the firing of nerve cells.
The promoter potassium channel combinations were tested both in mice and miniature brain-like structures grown in tissues, which were created using skin-derived human stem cells. We found that the immediate early gene was promoter, in combination with the TCNE1 potassium channel gene, proved to be highly effective in calming neuronal excitability following an induced seizure, and also the suppressing spontaneous seizures without having any negative effects on cognition.
Gene and stem cell therapies.
Gene and stem cells therapies hold promise for the treatment of a wide variety of inherited and acquired human disease. Identification of genes involved in human disease and develop of novel vectors and devices for delivering therapeutic genes to different tissues in vivo have resulted in significant progress in the area of gene therapy.
Isolation of stem cells from organs formerly thought to have no regenerative potential. The demonstration of stem cells plasticity, the creation of human embryonic stem cells clearly demonstrate the visibility of human stem cells therapy.
Much additional work remains to be done in areas of vector development and stem cell biology before the full therapeutic potential of these approaches can be realized. Of equal importance, the ethical issues surrounding gene and cell-based therapies must be confronted.